Hello everyone. I'll be sharing some of the insights and perspectives that we've gained on harmful algal blooms using in silico or computational mechanistic models. Harmful algal blooms, or HABs for short, are a phenomenon where certain species of micro and even macroalgae increase in numbers. These become harmful when the toxin from the causative organism accumulates through the food chain, or the numbers increase to the point that leads to oxygen depletion and consequently mortality of marine organisms. HABs causes many detrimental impacts. Illnesses and death can result from toxin poisoning. The Philippines, unfortunately, has experienced large numbers of paralytic shellfish poisoning illnesses, including 165 deaths. Socioeconomically, livelihoods are lost when HABs occur. Also, food sources for coastal communities are lost. Economic losses due to harvest bans and fish kills can be significant. One single fish kill can result in tens to hundreds of millions of pesos lost. The potential to export seafood is also compromised due to HABs. HABs leads to ecological impacts such as mortality of wild organisms and further deterioration of already impacted coastal waters. To help mitigate the impact of HABs, it is important to understand the patterns in bloom initiation, maintenance, and termination. HABs, though, are a complex phenomenon that occurs in the ecosystem through a multitude of factors. One approach towards understanding HABs is through the use of dynamic computational models. In this talk, I'll discuss some of our efforts to develop such a model, initially focusing on capturing blooms in Sorsogon Bay. Then I'll show how we use this model to conduct virtual experiments on bloom dynamics to test different hypotheses on bloom dynamics. Lastly, I'll briefly outline what we've done to enable easier application of the model to other sites. The HAB model combines physical, chemical, and geological factors with the biology of the HAB-causing organism and the toxin vector. The main HAB-causative organism in the Philippines is the dinoflagellate Pyridinium bahamense, which causes paralytic shellfish poisoning, and the main toxin vector are shellfish such as mussels and oysters. In particular, we developed the model to help understand the factors and mechanisms leading to Pyridinium bloom initiation, development, and decay in a tropical embayment such as Sorsogon Bay. One unique aspect of this model is that it incorporates key components of the life cycle of Pyridinium, with the cyst stage in the seafloor existing to become cells that can then divide or die or encyst depending on the spatially explicit environmental conditions they're situated in. The Pyridinium population model is represented using the agent-based model approach, where the agents or individuals in the model are its cells and cysts, undergoing the aforementioned life history processes depending on light, temperature, salinity, and nutrients. This Pyridinium model is coupled to physical models, namely a 3D hydrodynamic model of the site, including information on currents, temperature, and salinity, and also a watershed and diffusion model for nitrogen input into the bay. Light is derived from satellite data. The other unique part of this model is how it's coupled to an agent-based model of the shellfish, represented by the green mussel Pernaviridis. Here, the shellfish is the individual or agent that filters out Pyridinium cells within the localized space where it is located. Then, the transfer of toxin through two compartments in the mussel is represented, as well as their detoxification. This shows an example run of the model. This is a top view of Sorsogon Bay, where you can see literally down to the depths of the seafloor. Green dots represent groups of Pyridinium cells. Blue are groups of cysts, white dots are the mussel, and if they become red, that means their toxicity concentration is beyond the regulatory limit. Cyst beds are set based on the field data. They exist into cells. And during the early months of the year, the cells tend to be advected out. During the southwest monsoon, cells and cysts become more concentrated inside the embayment. And here you can see a mussel that several mussels that have become toxic. We ran the model many times using different combinations of values of the parameters as part of the parameterization and sensitivity analysis. We compare the different model results to 6 observed patterns to filter out the more appropriate model versions. The patterns are time series of cells and shellfish toxicity and spatial distributions from different field sampling months. These graphs represent 5 model versions with different parameter combinations or sets that more closely tracked the observed patterns, which are the rows in the bottom. The top plot is for pyridinium cell densities. This bottom plot is for shellfish toxicity. Most did capture the observation that the main bloom occurs during the mid-southwest monsoon around September, October, where there is increased rainfall. But toxicity can also prevail even after the cells have gone. This shows again the different model versions with different parameter sets and the observed data, this time for the spatial distribution of pyridinium cells in 4 months. Model version 117 was relatively better at capturing the observed spatial pattern. There were still some discrepancies. It showed the higher concentrations in September in the transition area from the shallow to the deep portion of the bay. Now, we have a model that could reasonably represent HAB models in embayments, where these events typically occur in the Philippines. We wanted then to examine certain mechanisms about pyridinium blooms. Specifically, we wanted to tease out the role of physical accumulation of cells due to water circulation and stratification, or the layering of the water column, versus nutrients from runoff, and how these interact with biological grazing. The context of this question is that typically blooms occur during the onset of the rainy season, and a common explanation is that there is an increase in nutrients from runoff. However, this could also be driven by cells being concentrated physically when stratification in the bay develops with freshwater input. Then, we were also interested in mortality processes such as grazing, since based on literature and the previous model results, this can impact the formation of blooms. The exciting part of modeling is how you can manipulate factors that you would never be able to do in real life. To investigate our questions, we implemented a HAB model stratification version, where there were different stratification scenarios for Sorsogon Bay. High rainfall leading to strong stratification and more potential accumulation due to less mixing in the water column. Moderate rainfall with weaker stratification, and low rainfall or no stratification, meaning more mixed water column conditions. Then we used 2 scenarios for nutrients in combination with those 3 rainfall and stratification scenarios. One is that regardless of the rainfall condition, nutrients were always high. The second is that nutrient concentration followed what was expected based on the rainfall. High nutrients for high rainfall and runoff, and medium for nutrients for medium rainfall and runoff, and so on. Lastly, we ran those scenario combinations with and without shellfish grazing. So what did we find? For the scenarios with varied stratification conditions but similarly high nutrients, they all resulted in high cell densities of pyridinium. But if the nutrient conditions follow the amount of rainfall, then the lone rainfall condition did not result in a bloom. This tells us that it is possibly the nutrients that drive the blooms rather than the stratification condition. An important point too is that incorporating shellfish grazing significantly diminished cell densities, although most likely pyridinium can still bloom in certain scenarios. Essentially, what we did here is to conduct a multifactorial experiment on the Sorsogon Bay ecosystem using this model. Sorsogon Bay pyridinium blooms are seen during the mid-southwest monsoon and it can extend through the northeast monsoon. Due to its life cycle, cysts are key in seeding the blooms first. Then the increase in cells appear to be driven by circulation that retains cells inside the bay, plus the influx of rainfall, which leads to stratification and, importantly, increased input of nutrients from runoff. Toxicity follows the bloom but persists even after bloom decline during the mid to the latter part of the northeast monsoon. Blooms are also mediated by mortality of the cells in cysts, such as grazing by the shellfish themselves, but likely by zooplankton as well, though we have not incorporated that in the model. This HAB model has so far been developed for Sorsogon Bay and Morcillagos Bay. However, HABs in the Philippines occur in many embayments that have their own particular characteristics that can potentially change bloom dynamics, and we wanted to be able to easily adapt this model to other sites. Thus, we have reconfigured this model to this end. Three key revisions were done. One is in the pre-processing of the input data. Input data for light, temperature, salinity, and current velocities can now be processed more easily from their original sources using automated scripts in R, and visualization of these inputs can also be done. For the main model itself, a centralized settings file was developed which consolidates the input data, allows tweaking of the parameters, and enables batch runs. Lastly, for the model outputs, scripts to automate generation of time series and spatial plots for the relevant information were created using R and MATLAB. Hopefully, we can apply the model to more sites and help further assess the similarities and differences in the bloom dynamics across the embayments in the country. So thank you to those who've watched this, and thank you to the many collaborators who've helped develop and analyze these models. And of course, thank you to our supporters, funding agencies.